Thank you everyone for attending the session today. I am Muhammad Raisul Abedin. Today I'll discuss about the efficacy of a small molecule Adipo 002 that promotes browning in human adipose tissue explant study. And our recent study have shown that the Adipo 002 molecule has promoted browning in human adipose tissue explants as well. I'm a senior scientist at Adipo Therapeutics. We are located in Indianapolis, Indiana. The research was partially funded by the state of Indiana through an innovation voucher grant program. To combat the current global obesity epidemic today, it is of great clinical importance to convert the energy storing white fat into more metabolically active energy burning brown fat. Induction of browning by the cold exposure or the receptor targeting has been shown clinically effective. The health benefit of brown adipose tissue include higher caloric burn, lower level of blood lipids, weight loss, improved insulin sensitivity, lowering the risk for type 2 diabetes and also other cardiovascular diseases as well. Over the past 10 years, we have investigated the efficacy of Adipo 002 small molecule in adipose tissue browning. Adipo 002 is a dibenzazepine molecule which belongs to the gamma secretase inhibitor class family that blocks the Notch signaling pathway. The Adipo 002 nanoparticle is a PLGA nanoparticle formulation that encapsulates the Adipo 002 small molecule shown in the red dots here. And the Adipo 002 nanoparticle is designed in such a unique way that has higher affinity towards the fat cells and that can be easily and readily uptaken by the fat cells very quickly, and also it can intracellularly release the Adipo 002 API. When injected in the fat tissue depot, the Adipo 002 nanoparticles are readily uptaken by the fat cells through endocytic mechanisms. The active ingredient Adipo 002 is released from the PLGA nanoparticles intracellularly over a week and in a controlled and sustained fashion. And this can actually transform the local white adipose fat tissue depot into a brown adipose fat tissue depot. Upon injection of the Adipo 002 nanoparticle, it gets readily uptaken by the local adipocytes at the injection site. When it is uptaken, the Adipo 002 nanoparticle formulation now can release the Adipo 002 small molecule, which is a gamma secretase inhibitor, and inhibits intracellularly the gamma secretase enzyme. This action blocks the Notch signaling pathway by targeting the Notch target genes like HES1 and HEY1 family genes. This results in the upregulation of the PGC1A and PRDM16 like markers, which are the classical biomarkers for browning. Treatment with Adipo 002 nanoparticles have confirmed the mechanism of actions of browning in fat tissues for both rodent model and also the pig models. The subcutaneous fat depot injections of the Adipo 002 nanoparticles, we have shown that it reduces the adiposity in the subcutaneous fat depot, and the treatment also upregulates the key browning marker gene UCP1 compared to the control nanoparticle treatment. This local action of browning also impacts systematically. It improves the insulin sensitivity, glucose tolerance in mice, and we have shown that the 4 weekly consecutive injections in the subcutaneous fat depot in high fat diet induced obese mice, it has reduced the body weight gain rate over the month. In pigs, we have shown that 5 weekly injections at the subcutaneous fat tissue depot, the Adipo 002 nanoparticle treatment induces the multiacular lipid droplet-like structures, as you can see here. And also, it also induced shown by the white asterisk, it also induced more mitochondria than the control nanoparticle treatments. And these are considered as the classical marker for browning. To find out a more potential translation of this treatment in human, we have investigated the browning efficacy of Adipo 002 molecule and also the Adipo 002 nanoparticle in human fat cells. For this experiment, we have differentiated the pre-adipocytes for 5 days in the lab. Once we got the matured adipocytes, we performed 2 separate experiments, one with the API, that is the Adipo 002, and another experiment with the Adipo 002 nanoparticle treatment. So these 2 treatments were done separately. The ADPO-002 nanoparticle treatment was done only on 2 days, like on day 3 and day 7, only for 1 hour of exposure time. But the ADPO-002 small molecule treatment was conducted for day 3, day 5, day 7, and day 9. What we have seen with this experiment is that the ADPO-002 small molecule, it downregulates the notch target genes HES1 significantly, and also at the same time it upregulates the Browning marker gene PGC1A with statistical significance. With ADPO-002 nanoparticles, the nanoparticle treatment in the fat cells, it also downregulated the HES1 marker genes, which is a key marker for the notch target genes. It downregulated significantly, and also at the same time it upregulated the Browning marker genes PGC1A significantly higher compared to the blank nanoparticle treatment. We have challenged our system, the ADPO-002 API treatment, with human ex vivo tissue explant study, and this is to confirm a more potential translation of this treatment in human. And we have conducted the ex vivo tissue experiments with 21 patients samples. The objective of the explant study was to confirm the key Browning marker expressions of PGC1A and PRDM16 in the human adipose tissue explants upon the treatment with ADPO-002. We worked with a very diverse demographics of patients. With the patients whom we worked with to conduct the experiments, the average age of the patients was 46 with a standard deviation of 11, and their average BMI was almost 46 with a standard deviation of 9. To talk briefly about the methods we followed to conduct this experiment, we collected both the subcutaneous and omental fat tissue samples from the patients who were undergoing the bariatric surgery. Most of them were going to gastric bypass surgery. And we brought those tissue samples right in the lab as soon as possible. We cut and minced the tissue samples into smaller pieces with 5 to 10 milligram range of weight. And then we transferred the all adipose tissue samples into an experimental culture well where we treated the fat cells with ADPO-002 molecule and also with the other control drugs as well. The tissue was harvested on day 7. We have extracted the RNA from the tissue samples, and with those extracted RNA, we further synthesized cDNA from the samples. Finally, we have investigated the expression, mRNA expression of the key Browning marker genes like PRDM16, PGC1A in those human adipose tissue explants. And we have used the RT-qPCR method to conduct the study. We have investigated the relative expression of the classical Browning markers like UCP1, PRDM16, PGC1A in the untreated tissue samples. We found that the UCP1 expression in both subcutaneous and omental fat tissue samples, the expression level was either very low or undetectable compared to the other two markers like PRDM16 and PGC1A. To avoid the complexity of this experiment with the UCP1, we have moved further with the PRDM16 and the PGC1A for our key Browning marker as our key Browning marker genes of interest. We treated both subcutaneous and omental fat tissue samples with the ADPO-002 small molecule. We have chosen 3 concentrations of ADPO-002 small molecule, that is 3 micromolar, 10 micromolar, and 30 micromolar. We have chosen forskolin as a positive control for this study, and also the DMSO treatment group was then chosen as a negative control. For the PRDM16 gene expression, we found that the 30 micromolar ADPO-002, it significantly upregulated the PRDM16 gene expression in subcutaneous fat tissue samples. And we found that the upregulation was almost 7-fold higher than the DMSO treated control group. For the PGC1A expression, we found that the ADPO-002 30 micromolar concentration, it upregulated almost 8-fold compared to the DMSO treated control groups, and it was almost very close to statistical significance. For the omental fat tissue samples on day 7, we found that the ADPO-002 30 micromolar concentration, it has significantly upregulated the expression of PRDM16 by almost 20-fold compared to the DMSO treated control group. For the PGC1A gene expression, we found that the ADPO-002 molecule, it upregulated the PGC1A expression in the omental fat tissue samples by 3-fold, and it also reached statistical significance compared to the DMSO control group. To summarize, we have investigated the efficacy of ADPO-002 and ADPO-002 nanoparticles in human fat cells in vitro. We have found that both ADPO-002 and ADPO-002 nanoparticles, they downregulated the expression of the notch target genes like HS1, and at the same time they have upregulated the key Browning marker gene expression like PGC1A. In the human fat tissue samples, we have studied with ADPO-002 small molecule, and we have shown that it upregulates the key classical Browning marker genes like PGC1A and PRDM16. In future, we are focusing towards more like the functional activity of the mitochondria upon the treatment of ADPO-002 molecule. We are right now investigating the mitochondrial density, mitochondrial function, and the role of UCP1 upon the treatment of ADPO-002 small molecule. With this, I would like to thank everyone who were involved in the whole study. I would like to thank the patients. I would like to thank the surgeons and all our colleagues at Indiana Bioscience Research Institute. And thanks to the state of Indiana for partially funding this project as well.